Okay, so upon ligand binding, we said that the ligand binds here, and that allows the G protein, this heterotrimeric, hetero, right, means different, trimeric, three, so because of the three subunits, so that's why it's a heterotrimeric G protein. That whole thing associates with the receptor, okay? So the next step in the process with respect to activation is when the G protein associates with the GPCR, the G protein coupled receptor, the affinity for GDP changes, and GTP replaces the GDP that is bound to that alpha subunit of the G protein. Now we think of this G protein as active, right? And so you can see here, GTP comes in and gives the boot to the GDP, right? And so there are some conformational changes that happen that allow for this and some affinities that change. But the end result of this step is that now the G protein is active, and after that, the alpha subunit dissociates from the beta gamma. Oops, dissociate. And they can both go on and mediate their own effects. I am struggling. Oh my gosh. Whatever, dissociate. I give up. Now they can both go and bind to downstream targets, and that's what you're seeing here, right? So here's the beta gamma that's going to go off and have some activity of its own, and the alpha is going to have some activity of its own, right? You can see it here associating with this effector. Now, that's the one category of G proteins, the one that use this heterotrimeric G protein rather than maybe just a single subunit. And so the second type, right, we talked about two types or two classes of G proteins. The second type are the monomeric, not heterotrimeric. Okay, so these monomeric G proteins function somewhat similarly, right? In this case, though, there's only one subunit. The first one that was ever identified is called RAS, and so that will be the example that we use, right? But now there are many, many more monomeric G proteins that have been identified. So our example here is the RAS G protein. And it's the same idea that typically inactive RAS is associated with GDP, ligand binding to the receptor, right, equals association of RAS that's ugly, but that word is association of the RAS with the GPCR. This allows GTP to displace GDP. All the same stuff that we've seen before. Not GTP, GDP. And now this G protein is considered active, right? And so now active RAS can signal in a variety of ways, many of which we'll go through here in the next couple minutes. Okay. It's important that these processes are able to be shut down. Okay, so we should talk, before we move on, we should talk about termination. How are they turned off? So the alpha subunits have inherent GTPase activity. So the alpha subunits are enzymes that break down GTP. Or in the case of small monomeric G proteins, the single subunit is a GTPase. So alpha subunit, or our example RAS, right, the monomeric G protein, has GTPase activity. So you can almost imagine that this activation is like setting a timer, right? And over time, that GTP will be broken down into GDP, which is what's shown here. And how quickly that happens can be influenced by things called gaps. Gaps are other proteins called GTPase activating proteins. And they're going to speed up the kinetics of the GTPase activity. And so you can imagine the gaps are going to be regulated. If the gap is active, it's going to turn off the GTPase act, or if the gap is active, it's going to enhance the GTPase activity of the alpha subunit, right? So the effect of the alpha subunit would be lower, right? So greater gap activity leads to typically a reduction in whatever signaling pathway. So we'll just say less G alpha activity, right? Because its GTPase activity will be faster. It shuts itself off, and gaps influence this. They speed up the process of how quickly these G proteins, whether it's the alpha subunit or here's the gap for monomeric G proteins, right? How quickly the monomer of the G protein breaks down GTP. Okay, I think I've belabored that enough. Let's move on to effectors. Let's talk about what happens when the alpha subunit is present and ready to do things, or the RAS is present and ready to do things, right? It's got GTP active. What happens next? So what do G proteins do once they're active? G proteins, whether it's the alpha subunit, the beta gamma subunit that's working together, or if it's the RAS, right, as our example, active G proteins tend to change the activity of effectors. What are effectors? Well, they're commonly enzymes that produce intracellular messengers. So I don't want to put my arrow here, but I want this idea to be that these effectors are commonly enzymes and these enzymes, when those enzymes are now active because the G protein has turned them on, these enzymes make intracellular second messengers. Okay, so that's the most common way that they work. Examples of this would be the enzymes like adenyl cyclase. That's clase, C-L-A. Another enzyme is phospholipase C. We'll talk about these, so don't stress. Okay, so these are a couple of enzymes that are commonly activated, and that's what's shown here, right? If we look at C or D, right? Here's inactive adenylyl cyclase, and we can see that it gets turned on and now can do something catalytic, right? So if this is off, none of this activity is here, okay? But you could imagine that a G protein, maybe a G alpha, right? Or maybe it's the beta gamma part, maybe it's RAS, right? One of these is going to turn this enzyme on, right? It's going to regulate this enzyme, and now there's all of this downstream stuff which we'll talk about. And the same is true for, if we look at D, it's the same idea, right? So here's an enzyme, phospholipase C. If this enzyme is off, it's just there, right? It's just present. But if the G alpha is active, or maybe it's the G beta gamma that's active, oops, that's supposed to be a gamma, or maybe it's RAS, right? We don't know, right, yet. We'll talk about which ones do these things, but upon activation, they target enzymes, right here, to cause change. And now phospholipase C, the enzyme, will be active, and all of this stuff can go forward, okay? Besides activating enzymes, another common type of effector, right, are ion channels. Right? So enzymes can be effectors, ion channels can be effectors, and that's what they're showing here in panel B. Right? That we've got some sort of ligand-gated, or excuse me, G protein coupled receptor. Well, I tell you what, let's hang on to B. How could an ion channel be an effector? Well, we can sketch it out, right? We can draw out the cell membrane, and we know that there's a G protein coupled receptor for some neurotransmitter in here. And upon ligand binding, if we've got a heterotrimeric G protein with an alpha and a beta gamma together, right, this will now associate, and that GTP will associate particularly with the alpha subunit, and now this thing is charged up and ready to go. So the result of activation of the G protein coupled receptor is alpha, and we'll draw this like, because it's active, it's going to look to do something, and beta gamma, which is the same way. And a target of this active alpha subunit of the G protein may be an ion channel. So for example, right, this can regulate ion channels. Okay? Association of the alpha subunit can change how the ion channel associates. A specific example would be not the alpha, but beta gamma can open up a certain type of potassium channel. Okay? That inwardly rectifying potassium channel that we talked about before is gated by beta gamma. Okay? So some G protein is going to help change this gate, and what it does is it allows potassium to flow. Okay? So another target, right? Doesn't have to always be enzymes, it can be ion channels. The book also gives the example of the alpha subunit associating with a voltage-gated sodium channel or a calcium channel, right? And making them harder to open, right? So that would have an inhibitory effect if we don't allow them to open as easily. So two different ways, two different targets, two different effectors for G proteins.